In this study, we looked at the neural correlates of confidence and accuracy during realistic decision making. We know that from the literature that it is possible to decode confidence or objective accuracy from neural signals in lab-controlled conditions with very synthetic or simple stimuli. What is not known, though, is A, whether these patterns are also present during a realistic task, and B, if they are strong enough to be decoded even in noisy environments. So what we did was designing a decision-making task in which participants were firstly shown for half a second an image of an imaginary word composed of two regions, 1 and 2. After those initial 500 milliseconds, we presented a second picture that was the real stimulus in which colored dots appeared on top of each region. Then the user had to decide whether the region that was most in danger was region 1 or region 2, taking into account both the number of dots and the color of the dots in each region. After making a decision, they had then to assess their own decision confidence in a range from 1 to 4. So our 8 participants were doing this task into an MRI room while we were collecting simultaneous EEG and fMRI data. The EEG was collected from 128 electrodes. The fMRI data was not used for this particular study, but the fact that the EEG was acquired inside an MRI room generated a lot of extra artifacts that simulate a real environment in which we would record EEG data. Our EEG data was pre-processed and bandpass filtered, and then we grouped these epochs on the basis of accuracy, so whether the person has made a correct or an incorrect decision, and on the basis of confidence, so whether the person has reported a low versus a high confidence for that trial. We then analyzed these epochs on the basis of the accuracy, so comparing correct versus incorrect, and we found that the neural correlates of accuracy were located into the occipital region bilaterally and peaking about 550 milliseconds after the onset of the stimulus. And if you think about this kind of makes sense, because in the first 500 milliseconds the user is still accumulating evidence and making up his mind about what to decide, and then in the next very brief amount of time, that's when the decision is formed, and those neural markers are actually the ones that are most informative to understand whether the decision would be correct or not. We then analyzed the epochs on the basis of the confidence reported by the participant, and we found that the neural markers of confidence peak at about 330 milliseconds after the stimulus and at about 900 milliseconds after the stimulus. So there are two critical times there, one during the presentation of the stimulus itself and one after the decision is formed, that basically kind of retrospectively assess the quality of that decision. So what we found in this study is that, well, first of all, the neural markers of accuracy and confidence are also present during realistic decision making and are strong enough to be decoded even in a noisy environment such as an MRI room. And second, that accuracy and confidence generate very distinct neural markers. These results are very important because they could then be used to develop brain-computer interfaces that could help us assess and understand how good we are in our decision-making task without requiring any feedback about the quality of the decision.